All right, y'all, that was a perfect fade out. Excellent. Thank you for joining us this afternoon for our Spring 2025 Applied Ecology Minors Research Symposium. Really excited to have everybody here. Really excited to get to celebrate all of the incredible research that y'all have done. So thanks in advance to our lineup of students who are presenting today, and of course to our mentors, without whom none of this would have been mentored, right? Y'all make a huge difference to our students and to our program. So thank you. Very excited to also share that as of this May, we have now graduated 182 students from the Applied Ecology minor. Give yourself a round of applause. This is my opportunity to remind myself and mentors that I have a collective resume that I send around. So I will be sending that around in case a student who you have mentored for the minor through their research has presented a poster, a talk. If you have published your work, we're trying to keep tally of all of those collective efforts and deliverables, because it's really amazing everything that y'all accomplish for the credits and beyond. Before we get started, I want to acknowledge that the lands within and surrounding present-day Raleigh are the traditional homelands and gathering places of many indigenous peoples, including 8 federally and state-recognized tribes: the Coharie, Eastern Band of Cherokee, Haliwa-Saponi, Lumbee, Meherrin, Occaneechi Band of the Saponi Nation, the Saponi, and Waccamaw-Siouan. We share an ongoing responsibility to safeguard these lands and to respect the sovereignty of the tribes and indigenous nations residing in North Carolina. Now, our goals in applied ecology are to study the world around us and apply what we learn to inform and empower stewardship. But it's important to remember that long before settler scientists decided this was important, expert knowledge and care for the world around us has been applied as an integral part of indigenous cultures in North Carolina and around the world. Acknowledgement is a form of decolonization, dismantling an existing power structure. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act, federal legislation that bought or forcibly stole land from indigenous tribes. The sales of indigenous lands raised $134,920 originally to fund NC State. That's the equivalent of $3,810,578 today. I hope that we can continue to disrupt and dismantle colonialism beyond a simple territory acknowledgement by supporting and empowering each of you as scientists. With that, I'm excited to learn from our speakers today. You'll see we have 11 students. So in the spirit of sharing our acknowledgements first, as we get Kenia Barajas-Salazar up to speak first, just take note, right? We've got 11 students and 11 incredible mentors represented today.
All right, so hi, I'll be talking about my research that I did over at the North Carolina Zoo in Asheboro. And so we were evaluating drinker use of hoopstock on the Watani Plains. If you're not familiar with the Watani Plains, it's over in the Africa side. And so it's like a 40-acre habitat with mixed species of hoopstock and rhinos. And so, these are my study subjects. So there are 7 different species of them. The water buck, the greater kudu, the Tonto gazelle, the Adro gazelle, the bongo, and then the rhino right there. And so in total, there were like 62 animals that have access to the habitat. And so for that reason, the main goals for the research was, do we need another drinker besides the big one open in the middle of the habitat? 2, evaluating how often the water hole was being used. And 3, if there's any aggression around the drinker. And 4, if safari affects space use of hoopstock for future research. And so these are just some images of some of the animals using the drinker use. And so there's the orcs, the bongo, and the rhinos. And so for methods, we use Zoom Monitor, which is a known application that's used across different zoos to collect data on their animals for welfare reasons as well. And so we collected 20-minute scan sampling from February to April. And so there was a total of 110 scan samples over 9 observation days. And so the questions prior to each focal session was if there was a safari on habitat and to the type of weather. So when it came to the safari doesn't start till April to October. And so once they started in April, if I was, so there's is 20-minute interval. So there's like 10 total scans. And so if I was more than 5 into it, then it was still no safari on habitat. If it was before 5, then I just deleted that session and inserted a new one with the safari on the habitat because it takes around 40 minutes for them to then exit the habitat themselves. And then for the weather type, it was just like if it was sunny or anything like that. And then for behaviors that we wanted to look was the drinker use and quantifying aggression, like direct aggressions versus indirect. And the way that we structured the ethogram for that was for indirect, like a head tilt, any like chasing behavior would see or displacement behavior where an animal will come in and the other one will move from that space. And direct behavior was just like physical contact with another species on the habitat. And so going into the results, I'll be first starting with the bar graphs showing the different behaviors. And so for drinker use by species, over on the y-axis is the average rate of drinker use per minute. And then on the x-axis, it's a raw number of observations per species. And we did the error bars based on the mean and standard deviation, so average rate of that. And so starting from the start, we could see and conclude that orcs are the ones using it the most, and then come rhino and the adra. There were 2 that were observed, which was the common water buck and the greater kudu, and that could just due to like needing more observation time with them. And so now is the indirect aggression between species. And so for this one, the same with the y and x-axis, we just didn't include error bars because there was just no variation for it to be included. There's a question where like maybe like the rhino or these have like more observations, but the reason because of that is because maybe when that observation was taken, there was more minutes in that one session for that day, and so those times varied, and so that's why even though the raw number is greater, the rate per minute is going to be greater, and that's where they're further into the graph. So before I go into like the heat maps that I produce with the space use per species, I wanted to go into like how the habitat map looks and what everything is. And so they have termite mounds, 3 of them located in the habitat. And then this is like the water hole here. And then the observation point is right there where I give you the majority of the habitat, just a few blind spots were just like when they went really close to the tree line where I wasn't able to see them. And if I was, it was just because then they like slightly moved over and I was able to see them with my binoculars. And then the safari road is highlighted in green. So for the heat map with all species with no safari and habitat, and this is more for preliminary data for them further research with safari on the habitat. This was located every 2 minutes on a heat map. So I will note down and press their locations for each one that I would see on the map. And so you can see that they use, when you're looking at it, an aspect of all species, in total, they use a large amount of the habitat in the majority. And then this is a number of observations with no safari and habitat. And so then when April started, we had the safari and habitat, and even though there's less observations, you could just see like they're not using it as much as when there's none on it. And so just dividing it by each species just to see like what the preference are on the habitat use. And this is where no safari on the habitat. The Adria gazelle uses a wide variety of the habitat from all like the left, right, middle, and the edges. And then the bongo are usually just stick to the right side of the habitat and then sometimes just move around a little bit and then come back to that area. Then the water buck, they restrict themselves to the right side of the habitat as well. And the fringe-eared orcs, There are some points where they're outside of the safari line, but majority of the times they're within that space of within that safari line, and they don't really go into a lot of the edge of the habitat. Then for greater kudu, the majority of their time spent is on the far left side or the far right side. And then for the rhinos, they just go all over the place. And for the Thompson gazelles, there's a lot of them, but they're like in small clusters, so they'll go in different groups. And so the majority of the time they're on the right side, but they do spend some time on the left side as well. And so for safari on habitat, again, keeping in mind that we had less observations with the safari on habitat due to it starting in April. And the only times they're on habitat is from 10 a.m., 11 a.m., and 12 p.m. And so when it comes to Aguirre gazelle, when there is a safari on habitat, we could conclude that they don't really move as much. They just kind of stay in one place, and that's usually on the left side. And then for bongos, they have access to outside of the habitat, like their holding spaces here, and so then usually they just don't come out as much when there's a safari out there. And then for the water buck, they just stay on the right side, and then when it comes to fringing oryx, it looks like they move a lot there, but they concluded like if they still stay within the safari line. But let's say like the safari is on this point, so they stay here. The safari comes right here and then move on this side instead. So they're trying to they definitely react to the safari truck in that way. And then for the greater kudu, they stay on the edges of the habitat there. And for the rhino, they still move around. I've seen them like chase another species while the safari truck is right there. And then for the Thompson gazelle, they still have like that same habitat space use when the safari is on habitat. So for the conclusion of the study, and this is a picture of the water buck there with their bushy tails. For drinker use, there was only 5 species that were observed, leaving out the other 2. And then for there were very few numbers and low numbers observed with drinker use and aggression. And this could be because the rhino or the Aguirre using the drinker use a lot more. When we're talking about aggression wise, because the frigid oryx, they're not really engaging in that, and they're keeping it like everyone away in a way that monopolizes it. But then right when they leave, then the frigid oryx comes in and drinks out of it. And then for the second drinker consideration, seeing as there's not a lot of usage of the main one right now, there's not a call of necessity to like invest in another one, but this can be furthered into like the warmer months as we're getting into summer for them to be more observations out there and then see if then the rate starts going up there. So out of the entire study, only 2 direct events was observed by the drinker, and that was between the Aguirre and the oryx. And so the Aguirre was the one that engaged into the direct aggression with it had bumped towards the oryx. And so this study was more just starting in space use, habitat use, with safaris in mind when they start and how they influence their space use, because they don't want to have the animals not use the entirety of the habitat just for like the tourist reasons. They still have the goal of the welfare of all the animals there. And so this is and just like thank you to like the staff at the zoo for all their help, and my supervisor Emily Lynch that couldn't make it today. Yeah. Any questions?
We do have time for a couple questions. If anybody has one to ask.
I have a question.
Yes.
I'm curious what their water availability looks like in the wild.
Mhm.
I'm sure it changes seasonally, but do you think like in the wild for that area, one watering hole would be sufficient for those species?
It just depends. I've read papers on it. There's always going to be like that natural hierarchy order with the water holes. But I feel like sometimes if there's like little availability in one water hole, the rhinos will like dominate that one. But if it's like available throughout the years, then they would be more tolerant of the other species around. Thank you. Yes.
You measure the rates of how often they.
Yeah.
Break. Do you have estimates of the body size of these? I would imagine that larger animals tank up, right? And have to visit less frequently, right? Smaller animals might have to, so like how the body size affect?
The animal care stuff definitely have like the sizes on animals. The way we did it was more. I noticed like on the warmer days they would drink more, especially the orcs and the rhino adra, and those were the ones that move way more than the other ones. And so the adra would were here for indirect aggression. The rhinos, they're like in the order of who would engage into it. So the rhinos and the adra were the ones that would engage the most with other species. And since they were like having more activity, then they were probably just getting more thirsty than the other ones as well. No problem. Yeah.
Thank you so much.
Thank you.
Thank you, Erin. I'm here today to talk to y'all about PFAS and gamma globulin. So first, what are PFAS? PFAS are going to be highly fluorinated synthetic chemicals. They will contain at least one fully fluorinated methyl or methylene group within them, and they're known to be very resistant to breakdown, regardless of if that is chemical, thermal, or metabolic breakdown. This has led to them being very prevalent in the environment and has given them the name of forever chemicals. There's an example of one PFAS up there called PFOA. PFAS, it's not just one thing, but many. The numerous studies have shown us that they are immunotoxic. Our lab in particular, the Boucher lab, has shown that populations of alligators and fish in the Cape Fear River Basin, those that have higher levels of PFAS exposure, are showing more signs of immune dysregulation. It has also been seen that children who have higher levels of PFAS exposure, when then vaccinated, will have a lower antibody response to those vaccines. So the big question for us is whether or not PFAS are going to bind and directly alter those antibodies. Is it changing properly? It was a little bit. Okay. So a little bit of background first between gamma globulin and antibodies. Gamma globulin is a very important part of your adaptive immune system. It's going to be all of your antibodies, and it's named gamma because when you take your serum proteins and run them through a non-denaturing gel, that first band at the top is albumin, your most common serum protein, but the second most common is going to be your gamma globulin, which is the last band on that sort of gel electrophoresis that we see there. So for my specific hypothesis in my experiments, I just wanted to test if PFAS bind to gamma globulin. In order to do this, I ran a differential scanning fluorimetry assay. This assay, I was using an environmentally sensitive fluorescent dye, which I call Glow over there, as well as a real-time PCR machine. So using these things, we will take gamma globulin and ramp up the temperature. As the temperature goes up, it will denature, and that causes a change in the microenvironment, causing the glow to then fluoresce, the machine to read it, and for me to make some graphs. So a closer look at this data with that blue curve there. This is the fluorescent response graph, directly seen as that glow is melting. The blue is going to be the melt temperature of gamma globulin when there is no PFAS associated with it. This is around 78 degrees Celsius. That's the normal guy. As we increase our concentrations of PFAS, the melt temperature is decreasing all the way to that orange line for our highest concentration. That is a 23-degree shift backwards in the melt temperature of your gamma globulin. So, kind of big. From there, I used the variable slope 4-parameter model, and from that, the simple cooperative model to create a concentration response curve, which is going to give me a KD. KDs are the dissociation constant, and that is important because the lower the KD, the stronger the association between your gamma globulin and your PFAS, which is how close they're interacting with each other. As we can see, the natural ligand of decanoic acid, this was a little bit of a positive control. All of our PFAS are the same, if not stronger associated than the decanoic acid for our KD. SEM just stands for standard error of mean. And our change in temperature for our max concentrations, those are not small numbers for a change in the melt temperature of something in your body. From that data, I can say that some PFAS are binding to gamma globulins, and in addition to being able to say something about my hypothesis, the binding of those PFAS, we can say, are destabilizing your gamma globulins. So this is actually kind of interesting in the context of research, since all of the other previous proteins we've tested PFAS with have had a stabilizing effect on them. But for some reason with gamma globulin, it's just taken it the other way and completely destabilizing the stuff. Future studies should examine if this destabilization is having a direct effect on your adaptive immune system or not, because it's kind of important to know. I want to do a huge shout out to my mentors, Dr. Scott Belcher, Zach McLean, and Dr. Mallory Thomas. It has been an honor to know these people and get to experience lab life with them, and I've learned so much from them. It's been incredible. So, are there any questions? Yes.
So gamma globulin is a component of the immune system.
Mhm.
Is it, I guess, represented at similar levels across different species, or do like alligators have more of it and mammals have less of it?
That is a wonderful question. So I'd have to say I don't have any papers that this information is going based off of, but anything that doesn't have gamma globulin won't have antibodies, and they won't have an adaptive immune system. And that just kind of means death as soon as you get an infection. So
I guess what I'm curious about is whether you would expect PFAS to have similar implications at similar levels across different species. Does that make sense?
it does.
So I was asking about the mechanism, but curious about the manifestation, right? Like what's going to happen to these immune systems?
I mean, we see it both in fish and alligators, which represent two different levels of an ecosystem, as well as people. So I'd have to say yes, anything that's in association with the water, because it is fully contaminated in your Cape Fear River Basin, by the way. Congrats. So anything that's in strong association with that water is just it's gonna be messed with. But
And
yeah,
is there, sorry,
questions?
is there like biomagnification of the food web?
Not in the same way that we see it with other things, because PFAS, it doesn't like fats, it likes proteins. Usually biomagnification is whatever's wrong or getting accumulated is in the fats, and then that keeps adding up. But because it's in the proteins, we just see it as an exposure level rather than as an up the food chain level. So
by accumulation, not biomagnification.
yes.
It's a way to field my random.
Thank you. Absolutely. Anybody else have questions? Yes.
Yeah, so it looked like when you were doing the study of dissociation constant versus the change in the melting temperature,
Yeah.
there wasn't exactly like a clear correlation there. So is there any idea what is causing that change in the melting temperature if it's not that dissociation constant?
Absolutely. So for some of these, we could only get the concentration of the PFAS up so high before it just fell out of solution no matter what. So for something like PFOA, you have a very good concentration, dissociation constant, but we couldn't keep it in solution for very, very high concentrations like we could for PFHXA. So it's delta TM for the maximum concentration we could get. Doesn't look as big as what we could reach for PFHXA. Absolutely. Any other questions? Beautiful.
One more.
Yes.
So if you have these binding efficiencies, does that mean that there's potential to have like a gamma globulin inspired filter that could, you know? Here's a patent for you, right? Because I mean, you essentially, you could make a seine or a filter and
Mm-hmm.
could it bind it more?
No familiarity and possibility to answer that, so if I'm not butting in.
Go for it.
So albumin is a primary binding protein that transports PFAS all over. That's where we first started studying. It's really complicated. It's got like 8 different binding sites on the same molecule. And so these types of studies get really complicated. So people have tried transfusions and other sorts of things, which appears to at least temporarily lower it, but really it's not an answer to a industrial pollution problem. So
Turns out.
those comments aside, but some things like that have been looked into. I think here, what's really important is that destabilization and that potential direct effect, which nobody has looked at on the antibodies themselves. So this is some really exciting data.
It sure is. Awesome. Thank you.
All right, hi, my name is Ben Kreiser, and my research was on living shorelines and fish down at Seamast, which is in Morehead City. So first off, a little bit of background on living shorelines. You know, they're made of natural materials. They allow for natural coastal processes to remain. They provide erosion protection. They're very resilient against storms. And one of the big features of them is that they attenuate wave energy instead of reflecting it. And because of this, they are considered like soft shorelines. And because coastal erosion is such a big issue, there's been a lot of research on the effect of living shorelines, and 2 of the more relevant ones is that they provide nursery habitat and trophic support, and that living shorelines can show improvements in nectar communities, but that improvement can take time to develop. And so my study site was located at Pine Knoll Shores, specifically the aquarium, and this is just a brief timeline of living shorelines at Pine Knoll Shores. And the most important thing to come from this timeline is that the stone sill was installed in 2001, while quick reef and exoform shorelines were installed in the fall of 2024. So my research question was, how does fish utilization of living shorelines vary by substrate and time of year at the Pine Knoll Shores Aquarium? And then my hypothesis was that the natural shoreline or the granite sill shoreline would support higher fish abundance throughout the survey. In order to do this, I did 20 minnow traps, 5 of them per shoreline. All of them were baited with cat food. The cat food was placed into these clear vials that had holes drilled into them. And then traps were placed during a falling tide and then retrieved the next day, which was also during a falling tide. And this allowed them to be left out for a full tidal cycle. The traps were placed approximately 13 meters between them and also any other structures on the shoreline. And the traps were placed 1 to 3 meters away from the living shoreline, and all of them were anchored into the sand of stakes, like the picture you can see me doing right there. There are 4 shorelines that I sampled: exoforms, quick reef, granite sill, and a natural shoreline. And I collected data on species counts and the lengths of the first 10 of a species. So this is just an image so that you can see what they actually look like. On the left is the exoforms, which is formed out of concrete, like think of a mold of some kind. On the right is a quick reef, which is calcium carbonate blocks with like oyster shells embedded into them. On the left is the granite, which is just chunks of granite. And then on the right is the natural shoreline. Pine Knoll Shores is the red dot dot on the map of North Carolina, and then the red dot on the inset is the approximate study site, and then the map on the right is each of my trap locations. These are a bit more high resolution, and the blue traps are the quick reef, when traps 1 through 5, yellow are the granite 6 through 10, and the red are the natural shoreline, traps 11 through 15, and the purple are exoforms, traps 16 through 20. In over 3 trapping sessions, 228 fish were captured, with 6 species observed. Those were the mummy chog, striped killifish, naked goby, pinfish, one unknown, and an inland silverside. The top left is of a mummy chog, top right is of the striped killifish, bottom left is of the naked goby, the bottom middle is of a pinfish, and the bottom right is of the unknown. My results, the preliminary data seem to support that the granite substrate had a higher abundance of fish throughout the survey. And over the entire survey date, I captured more fish in the month of March. But after performing a two-way ANOVA test, I found that there is no statistical impact of time or substrate on fish counts, which means that there's really no difference in the utilization of living shorelines by fish at the Pine Knoll Shores Aquarium, which does not support my hypothesis. The main challenge experience was potential conflict with other experiments. There was a large section of natural shoreline in the red oval, and I was going to place my traps there, but along it were hydrophones, and placing my traps there would have potentially caused conflict with their data. And so I moved them to the current location. There were multiple potential sources of error. The bait being used is the one of them. It was cat food, which may have selectively attracted certain types of fish, or it just may not be as effective as a different bait type. The ages of substrate is another factor, as the quick reef and exoforms, they are similar in age, and even then they have matured in slightly different ways. And then the last one is trap selectivity. The traps I used were unable to catch all of the fish that I observed, because multiple days when I was out setting and retrieving traps, I saw mullet swimming in big schools swimming around, and I never caught any of them in my traps, even though they were swimming in the same area that my traps were. Future research, it would be really interesting to sample these shorelines for an entire year, as this is just 3 months and it's really not indicative of the entire temperature range of a whole year. I would also like to see a study done, a before and after control impact study done with these shorelines, because they are especially exoforms. It is a relatively new type of living shoreline, and a before and after control study would be a lot better at determining if they show any overall improvement in fish abundance over a natural shoreline. And some more besides, a very big thank you to my advisors, Dr. Stacy Zhang, Dr. Trevin Toon, and my field assistants, Georgia, Vanessa, Gianna, Braden, and Sarah. And any questions?
Oh, you have a question?
Yeah.
From David Wagner.
So with your mentor that you were working with, did they also have a like their own hypothesis as to what they thought might be the best substrate? Like, was there one that you guys went into thinking separate from what you were looking for?
Are you asking if both me and my mentor kind of agreed that the granite would support higher fish?
Yeah, and I guess further, since you were working around the aquarium, if they also had their own idea too, if you encountered that at all.
I think we were generally on the same page about it because, like I said, the in this photo you can see that where I'm holding up the trap, you can see how much algae and growth has occurred on the granite sill. Whereas in the photos of the exoform and crick reef, there hasn't been a lot of growth yet. So we kind of agreed that we were thinking that that or the natural shoreline would support a lot more just on how long it's been out there and how much time it's had to develop.
Yeah, we have another question.
So, good stuff, Ben. You looked at abundance among the different types, but did you capture all the different species equally across the different types?
No, we did not. Mummy chogs were the primary species that we caught. Particularly so back to the two graphs, the one graph that shows the incredibly high count for the granite and in March, that was because of the photo of me holding up the trap. There were 81 mummy chogs in a single trap, which really skewed that specific time point a lot. So that's what kind of contributed. And so yeah. Also to that point, the first two sampling dates were primarily mummy chogs, and then the third sampling date, I caught fewer mummy chogs and a lot more pinfish. So I don't know exactly why. I would like to do more with it, but
Do you think you'll have opportunity to use different types of traps? And
yes.
how do you think that would change your results?
Yeah. Some of in one of the papers that I cited, they used nets with much smaller mesh size, and they're likely able to capture a wider range, more fish and stuff. That's kind of part of like doing the before after or sampling for an entire year. I mean, putting out nets would be allow me to catch a wider range, more mobile fish, because mullet, they don't really hover around structure as much as like pinfish and mummy chogs really do.
Awesome. Any other questions? Thanks very much, Ben.
Hello, I'm Jordan Holcomb. I am a junior. Oh, that's weird. I can see my words. I'm a junior in plant biology with a minor in applied ecology. And I did an experience at the Yates Mill Aquatic Conservation Center on if snails prefer live plants in captivity. How do I move this?
Click once with the trackpad. That should then enable you to do it.
There we go. So the species at the Yates Mill Conservation Lab is the magnificent ram's horn, and you can see them here in this little bucket. This was me transporting them over to my experiment. They were completely taken out in the wild, but this guy named Andy Wood, he brought some into captivity to save them from a hurricane right before the last ones were taken out. And he kept them alive in a sanctuary at his home until NC State and the Wildlife Resource Commission got involved and started holding other captive populations. I think it was in 2012. So props to this one guy who really saved this whole species. Now we're working on getting them back up. So in 2023, there were 2,000 individuals that were released back into the wild. It took a lot of preparation to do this, mostly with the establishment of aquatic plants and water quality. And if you came to this last semester, I did another research study on water quality and found significant results of water quality impacting the success of aquaculture. So I was like, why not look into the other factor that they're doing and see if there's any impact of aquatic plants? Because in the lab, they were only rearing them with artificial plants. And so I thought, if you need live plants in the wild, do you need live plants in captivity? Well, this helps them breed better, lay more eggs, make them happier, all of that. Okay. So I hypothesized that they would prefer the live plants because it just made sense to me that they would prefer something that's alive rather than something that's made of plastic. I wanted to make sure that they weren't using the plants as a food source because in the wild they do eat the plants. So I made sure to keep them very well fed so that that wouldn't be a factor. You can see these are their plastic plants in their very dirty tanks because the snails are actually pretty gross. Um, and I was thinking these snails have all been reared completely in captivity. They've never been in the wild, so they've only ever known artificial plants. They've never seen a live plant. So will they have like an instinct to want to go on these, or will they just prefer what they've always known? Oh, and these are the eggs. I don't know if you can see them very well, but these are their egg clutches. Like I said, I wanted to remove all the variables like food availability so that they wouldn't resort to eating the plant matter. And I wanted to make sure that oxygen wasn't an issue, so I made sure that the air stones were uniform throughout all of the different tanks. I used spatter dog plants because that is what the Wildlife Resource Commission sent us, and that's what they use in the wild when they're restoring these habitats that those snails are going to be released in. And in the wild, they do use these plants as egg laying and a food source. They lay on the bottoms of the leaves. And I wanted to make sure that the the live and artificial plants were the same type. So I made sure that the artificial plants had similar leaf shapes and size to the live ones. So I put one of each of the live and artificial in varying placements in these tanks. The green is the live and the red is the artificial, and the star in the middle is where I put the air stone. And I also fed them on top of the air stone so that it could be equally dispersed throughout the tank, equidistant from the two plants. I had the two opposite sides on the bottom, and then I put them one right behind another in the top tank. There were the 3 replications, obviously, and I put 20 snails in each. I counted the snails if they were on the live plants, the artificial plants, or neither. And if they had laid any eggs, I collected the eggs and moved them into a basket for them to hatch because I didn't want to have the babies in there being another factor. I didn't originally think that they would be laying that many eggs on the tank because they usually lay so many on the plants. But after the first count, there were a good number of them that were not on live or artificial. So I added that 3rd category of on the tank. And I cleaned them Monday, Wednesday, Friday, and I fed them, like I said. So my hypothesis was supported. They did prefer the live plants. There was this initial dip, which is really interesting to me. I don't know if that was because maybe they didn't know what the live plants were yet because they'd never been around them. So the first count I did, they preferred the artificial plants, but then it significantly changed after that first count, and the rest of the study, they preferred the live plants. And that was from just counting the snails themselves. When I counted the eggs, there was not a significant difference. At first glance, it looks like it, but as you can see, the error bars all overlap. It was due to the variation between weeks. It could have been maybe I didn't find a clutch one week, I found it the next week, and it messed up that. But it's kind of difficult to make sure I got them all. I tried my best. They're clear, so I was feeling around. Yeah. So, wait here, I'm gonna add this. So the main point of what I wanted to do was see if they were laying more eggs if they had live plants, because the whole point of this lab is to get them to lay as many eggs as possible to get the numbers up to be re-released into the wild. And they didn't seem to like the live plants other than for their mental health. They did not lay any more eggs on the live plants. So there were these pest snails called physids. You can see they're the little pointy guys, and they can outcompete the ram's horn snails that we were trying to raise. So I took them out every time I saw them, but as you can see, they're crazy and their numbers pop up really fast. Somehow they get through the filtration system. We found the eggs on the UV lamp before, which doesn't make any sense because the UV lamp is there to kill everything, and somehow these guys make it through that. So I wanted to add that because there was a little bit of competition between the ram's horns and the physids. Even though I tried my best to get them all out, it did happen occasionally. They also laid eggs on all the plants. I didn't count their eggs because I don't care about the physids, because they're bad. And the whole point was, should ramshorn be reared with live plants going forward? And after this experiment, we created a mock pond. You can't see the plants very well, but there are 3 of them in here. I made a mock pond for public education to tell the public what the goal is about re-releasing the snails, because they do tours of the lab, and people can see the snails, but it means a lot more if they see them in their natural environment. And like my first data points said, apparently the snails like the plants, they just don't lay more eggs. So these snails in this mock pond are just really happy, I guess. When I was counting, I did notice a correlation between the size of the snail and the size of the leaf, which is really interesting. It was too much to actually like track that. I didn't want to change my study halfway through, but that was very interesting to me. And after we finished this study, we started another study on calcium dosing to increase the strength of the snail shells so that they can be stronger when they're released. And that study has had really promising results. We haven't finished analyzing them, but the calcium has really been improving the snails. So that's exciting for looking forward. And my mentors, Chris is the head of the lab, thank you. And Loretta is the snail lady that taught me everything about snails. Any questions? This is an endangered fish that they rear. Okay.
For the visit day guys, did you see any like correlation with where they were laying eggs? Like, were they actually starting to lay on the live plants once you introduced
I
them more?
didn't count the eggs of the physids because I was really trying to get rid of them.
Yeah.
And it was more just like kill the eggs. But they just kind of lay so many eggs that they're just everywhere.
Just wondered if maybe they had a preference, like more so than the ramshorn for the live plants.
Yeah, I would love to look into that.
Take that
Yeah.
area from them.
No, like preemptive competition.
Yeah, that's a good
Because they're
point.
already more abundant and they seem to rear faster too.
Yes,
Just-
they could definitely outcompete them. I wish they were the endangered ones.
Well, then you'd have to help.
Yeah.
I have the same question. for you.
Okay.
Skylar Hopkins is on soon. She asked, she thinks she might have missed it from your methods, but did you control for the amount of surface area available for the real versus fake plants?
I did that by making sure that the real and fake plants were of equal size when I started, plant size and the leaf size and shape. I like got special spatter dog artificial plants so that they would be the same. You can go.
If they really like the glass, maybe they just really like flat surface. It's maybe not the shades or whatever.
Yeah, well, in the wild, they obviously don't have the glass, but the leaves are their flat surface. So, because I had thought of that,
Super spinning
I guess,
plants.
but
They might not.
they just need a flat surface. But that makes it weird that they hung out on the live ones significantly more than the artificial ones, though, because why are they doing that? Go ahead.
This is more of a general question. Does
Okay.
anybody look, so now, there's a great story about how they've survived,
Yes.
but now they've been reared for multiple generations
Mhm.
in a plastic-oriented system,
Yes.
which is a hard thing, and we study endocrine disruptors. I'm wondering if in your thinking, this is maybe there could be
Mhm.
plasticizers that are
Mhm.
influencing things, changing, sex determination in these guys.
They're hermaphroditic, so it doesn't really matter.
Okay, so there's my.
Yeah.
ignorance shining. But do you think that, there's room for the hypothesis that actually maybe there's a repulsive effect of maybe chemicals that are being leached from the plastic?
I think that is a really cool idea. I don't think that my lab will actually do a study on that though. I do think that's a really good idea. And I had kind of thought of it as why not give them live plants? Because they're hypothetically going to be healthier for them because plastic bad, but didn't really make a difference.
It's hard avoiding regrettable substitution.
Yeah. Well, and most of their tanks, a lot of them are plastic too. Some of them are glass, but some of them are, the ones that they live in regularly are made of plastic, and the ones I did my experiment in are made of glass. So I don't know if that had a difference.
Maybe the future is plastic, right?
I'm also curious, since they eat the live plants, in the wild, I'm curious if maybe every once in a while they eat the fake plants and if that affects anything. I don't know.
Dr. Kathy, did you?
Yeah.
Go ahead.
I think I heard you mention their mental health.
Yeah.
I just I love hearing that.
Oh yeah, that was kind of a joke because they it had no effect on their reproduction, but they like the plants. So I guess they're just happy. We love happy
I
snails.
love hearing that.
Go ahead.
Yeah, following up on Scott's question. So what you're looking at how you introduce them and then you monitor where they end up over time.
Mhm.
And so that's sort of a classic choice type experiment.
Yes.
And I have a model basically shows that if you look at the early time period, that's sort of a measure of colonization,
Mhm.
preferential colonization. And if they have preferential leaving, it tends to accumulate over time.
Mhm.
So perhaps
Which-
what you're seeing, maybe seeing preferential leaving
Mhm.
of the plastic.
Mhm.
Of glass, and I relate to what he's
Mhm.
saying.
That's a really good point.
I need a few more replications.
Yes. And I didn't mention this, but I did it over 12 weeks. So it was a pretty significant amount of time. Yeah. Anything else?
I just wanted to add that.
Okay.
That early part is so cool and interesting.
I know, this is like the
With
coolest
your
part of the
happy
whole thing.
snail analogy, I kind of like this because it links into behavioral habituation that you would see, for example, in a mouse behavior study in the first 5 minutes of a novel introduction.
Mhm.
So
Yeah, when I got my first data points, I was like, oh darn, this is gonna be a bad experiment, but then worked. Yeah. Anything else?
Thank you so much.
Hi, my name is Marley Kelly, and I was interested in seeing if there was a relationship between biodiversity and the value of single-family homes. So, oh gosh,
Like ones with the trackpad and then, yeah.
there we go. Okay. So in terms of like general ecology, this relates to landscape ecology within urban areas. So in urban areas, there could be patches of green space. There can also be patches that are just concrete, like parking lots and things like that. And a lot of these green places can come from residential areas and homes that have yards and things like that. And in literature, there has been a correlation found called the luxury effect, in which urban areas with higher median income have a higher level of biodiversity, both with plants, and there have been some studies that have found it with birds. So what I wanted to look into was, instead of income, the value of the properties and the homes in the urban areas. So I was looking at single family homes. So yeah, that's what I wanted to look into. And what I thought would happen is that with increased tax and market value, there would be an increase in biodiversity. So for this study, I used data collected by the annual triangle bird count that Dr. Kati facilitates. So in the triangle, there's a bunch of different survey points that people can volunteer and sign up to survey birds at. And they basically just stand there and count how many birds they see or hear. So we had data from 2019, 2021, 2022, and 2023. And the first thing I did with this data was I put all of the bird count points in a GIS map, and then added a Wake County subdivisions layer and select by location to get points that were within subdivisions. And then I also added Wake County addresses and selected them by the subdivisions to ensure that they were not like a business or something like that, like a residential home. And then I went on an application called iMaps through Wake County government to ensure that the addresses were single family homes and not like rentals or apartments and things like that. And then I narrowed the points to within 100 feet of the homes. So a lot of points were like this. Most of them only had one house, but a couple of them had 3 houses. So with the data, I averaged the tax and market value of the homes if there were multiple. So next I input all of this data into Google Sheets with Zillow data on market value and tax value of the homes. And this was based, all of the values were estimated in April of 2024. And then I also gathered the species richness and abundance measurements from the previous year's data and associated them with each point that they were associated with. So that was all together. And unfortunately, we don't have a lot of data on all of these points because when people are signing up for their points, they'd like to be in like the fun nature areas and not like next to somebody's house when they're trying to find birds. So I only had 22 points, and that may have affected the data. So I did a couple graphs comparing market value and tax value with species richness and abundance. So in all of these graphs, you can see kind of a negative trend with home value, which is surprising. But when you look at the R-squared value, it's very low. So that basically means there is no correlation. And this could just be because of the small sample size. There also seems to be a lot more samples in the lower value range and not as many in the higher value range. So it's really just, we need to do more data collection to see if there's actually a correlation to be found. And when I started this study, I wanted to look into resident-owned versus rental residential areas to see if the ownership of the land had anything to do with how much biodiversity was there. But it's really hard to get renter information and things like that. And I didn't have time to do that. But I think it'd be something really cool to look into, as well as homeowners associations and their regulations with yards and things like that. And then I thought it would also be interesting to see if homeowners plant plants to attract birds, if they care about attracting birds to their yard, or when they're buying a home, do they care if there are trees outside, or if they see a bird outside, they're like, oh, I like this house, I'm gonna buy this house. And things like that. And even though I didn't get great results from this, it was still really fun to be able to look through the data and do the data analysis process. So I want to say thank you to Dr. Kati for mentoring me through this project and helping me decide my research question. And also thank you to Dr. Peterson and Hannah Desroche, who is a PhD student at North Carolina State University. They helped me get started with research, and I assisted Hannah with one of her papers and was able to present that at the NCTWS conference this year. So yeah, thank you to all of them, and that's the end. Any questions? Yes.
I'm curious, so many of the, especially newer neighborhoods, Siegle and Lake County seem to be adjoined townhomes, so you lose that buffer property, the green space around the foundation of a single home. And I was curious what effect you think that might have if, yeah, I guess what was the representation of those types of houses within your data, and what would it have looked like, do you think, just hypothesizing, if you had more of the standalone houses with, yeah, just more, at least a little bit more space around them.
Yeah, I think that's definitely something interesting to look into. I didn't look into that specifically, but I imagine with especially the more expensive home, like there's a couple like $1 million homes. They would have a larger area around their home. They wouldn't be right next to someone. But also like at my townhouse, there is quite a lot of green space just because there's a yard in front of everybody's house. So even though it may not be around the whole property, it might be more just because there's more houses. But also that goes back to the ownership and landlords control the yard, and you can't just plant whatever you want if you're a rental. But in your own house, you can plant whatever you want. So yeah.
For the higher value homes, did you find more often than not that they were high value because they're newer and more developed, or that they were historical and they had been there for a while? Because I know in some places like North Hills, you'll see houses that have been there for a long time, that they're expensive because of location and because of how long they've been there. And I wonder if like that lack of disturbance happening there allows more things to be there.
Yeah, that's a good point, especially with the amount of construction that's happening in Raleigh and how much it's growing. I'm sure those homes are more left alone. I think when I was looking in the tax value data, a lot of the homes were older and had been owned for a while. So I think it may be because they're older, but I didn't specifically look into that. Yeah.
Have you gone to participate in one of the bird counts yet?
I'm not. I'm gonna do it this year though. So, and we need more volunteers. So if you're interested, let us know.
It's active right now.
Yes.
It's a really simple
Mm.
question that may or may not influence anything. Have you tried alternative fits besides the linear fit? Because
I have
that
not.
looks like a not or fit to me. Might give you a better fit to some of your data.
Yeah, I think when I was messing around with it, we tried like a quadratic fit and that looked interesting somehow.
Because I tell a lot of my students, we only use linear fits because of history
Mhm.
and graph paper. Now that we have computers, we could probably largely do away with linear fitting. So.
We'll speak on that for you.
Yeah.
So I wasn't directly associated with the project, but I helped look at the data. Looking through it, I said, hey, do that quadratic fit, looking at it. We didn't have time to go through like linearizing the data, see what that would look like. I think again, some of the issue was just the lower sample sizes. We didn't want to overfit the data. We didn't want to say, oh, look, here's a 100-degree polynomial that fits this perfectly. So it's, yeah.
Yeah, I didn't have a lot of time. I definitely want to look into different associations there might be. But this was just kind of like a preliminary.
Your left end of your curve said, thinking that
Mhm.
might not be a linear.
Yeah, we tend to get these sort of triangular spreads quite often. I think one of the things that this is just looking at one variable.
Mhm.
Right.
It
There's
tends to be
a lot.
Always a multivariate context in urban areas, so that would be the way, that's what you need to do, is looking at multiple variables.
Mhm.
So if there was a nonlinear process related to value, how would you hypothesize it would work?
Maybe the more medium value homes are higher density, and like the lower value and higher value homes are off towards the edges of the city. I'm not really sure. It kind of relates back to green gentrification and things like that. More green areas will become more expensive, but there are some areas that look kind of neglected that are still green, but not like the pretty green that everybody wants. So maybe that could be something with it. Do you want to?
Yeah. I think that there may be more variation in some parts of the spectrum rather than elsewhere. Depending on, which could be because of the variation in location to some extent and what the neighborhoods are like, as well as people's flexibility in being able to do one thing or the other. Whereas higher end or there might be sort of market influences in terms of maintaining values of homes, keeping them appearing in a certain way. Not here, there's a lot of potential factors that can affect how individuals make decisions for their homes. So in a sense, that this hasn't been at the level of individual home decision making, there isn't a lot of work on this. So it's something that we're curious about to see how market value intersects with how people make decisions. Because previous luxury effect work has mainly looked at income, which is a census
data. But so that is like 5 or 10 year intervals, and this is market value you can track more closely. And just to digress a little bit, but one of the things that I'm curious about is the way the market has changed dramatically in the last few years. What is that doing to the landscape?
My sister caught like a new house and like there was a lot of trees in the backyard, and then she was saying that she's been caught a lot of them. So there's so many birds right now. They'll cut in like the shade. So then she like only cut a few, just like for safety measures. I was like, okay, and because I live at a bridge where I live right now. And then front there are some trees, but they only cut like one tree like halfway because I was like, no, I like that tree. But it's like funny like how but I'm interested like in wildlife, but like maybe these ones just don't care as much. But it's interesting that we're looking into that.
Yeah, it'd be interesting to see if people care about the birds or if they just don't think about it when they're making decisions about their home. Yeah.
It's not a question mark. Just like, yeah, like what you're saying. Related to this, like green stormwater structures that help like prevent flooding and like water quality, and also like create higher value in areas, are also like associated with like higher health.
Yeah.
So like those green spaces, there's a lot of them have like I guess like a stormwater like functioning stuff, and I wonder if I'm not asking you, I'm just saying, but could kind of like interact with it.
Yeah, that'd be interesting to look at as well.
Awesome. Thanks, y'all. Thanks for the question.
Hi, everyone. My name is Sarah Siegle, and my project was using machine learning to assess trends in phytoplankton populations. So, as a background on this issue, cultural eutrophication is when you have the over-enrichment of nutrients in waterways, which can lead to algal blooms, and this is human-caused. So, in the Neuse River estuary in North Carolina, this is a major issue and a major issue around the world. And it's caused by this nitrogen and phosphorus pollution from urban development and also agricultural operations like the manure and fertilizer that run off into the waterways. And this can cause major fish kills, wildlife death, human health problems like gastrointestinal issues, neurological issues. So it's a huge issue in our North Carolina and around. And as you can see, this is devastating to fish populations and can have some pretty negative effects. So a bit of background about machine learning. It's a form of artificial intelligence that is very useful when dealing with large data sets, and it can use pattern recognition through the use of modeling approaches called algorithms to identify trends in the data. And the version I used was WECA from the University of Waikato, and it's a very iterative process. We try a bunch of different algorithms. You're constantly manipulating the number of attributes you're dealing with. So it's a very iterative process to get the best model possible. The main objective of this study was to examine how environmental and seasonal factors are affecting phytoplankton populations over the long term, as the dataset I used ran for 26 years. And the two main research questions I had under this are how are phytoplankton species varying with the seasons, and how are phytoplankton species affected by specific nutrients. The data set I used was from the Neuse River estuary, part of the larger Albemarle-Pamlico estuarine system, which is the second largest on the US mainland and the first in fish nursery ground on the US Atlantic coast, which shows why this is such an important area for research to be conducted. The data set I used was from 1994 to 2020 through biweekly to monthly monitoring, and it was classified by season. And the data points I had from this data set were phytoplankton species. There's about 100. Then we had nutrients like nitrogen and phosphorus, and environmental factors like temperature, pH, salinity, and dissolved oxygen. And this all led to a very complex data set with about 30,000 data points, which is why machine learning was very helpful in identifying patterns in this data. So when trying to find out which model best fits our data, we use 3 different statistics or metrics to pick out this best model. And 4 algorithms of these modeling approaches were tested, and they're each verified by 2 different methods to make sure the machine learning model is accurately representing the phytoplankton and environmental factors. And we also used it to detect correlations between the specific nutrients and the phytoplankton. So here is a graph that shows the performance of the 4 algorithms that we tested. The top green line represents random forest, which performed the best and best represented the actual data of the environmental and phytoplankton factors. And at its peak, it had over 95% correctly classified, which is very, very good. And as I mentioned, machine learning is a very iterative process. So we went down in the number of attributes, and most of the algorithms peaked at 41 attributes. So this is an example of how the machine learning makes its patterns and classifications in a decision tree. It starts at the top with temperature, and on the right, there's the higher temperatures, and on the left, there's the lower temperatures. And then in the branches, it goes down and classifies by nutrients, taxa, and finally by season, which is what we are classifying by. Now, in terms of the major correlations that the machine learning identified, we found correlations with nitrogen and phosphorus and specific species. The ones in bold were the strongest associations, and all of these, almost all of these species are potentially harmful species, harmful blooming species. So that's concerning, because if these are the species that are correlated with nitrogen and phosphorus and can cause the algal blooms, this is not good if we have extra nutrient pollution in the waterways. So in other words, to recap this correlations that I just showed you, phosphorus is correlated with gymnodinium and Oxyrus marina. Nitrogen is correlated with gymnodinium again and Heterocapsa rotundata and Provocentrum minimum, and ammonium is correlated with Wisteria. So this means that these potentially harmful species are correlated with nitrogen and phosphorus in our waterways, which should hopefully inform how we're limiting our nutrient pollution the best we can. Because climate change is a process that happens over the decades, it'd be interesting to see how the long-term changes in these phytoplankton species are occurring with a change in temperature. And as the harmful phytoplankton species are known to thrive in warmer conditions, especially with nutrient pollution, it would be interesting to see how these species compare to the beneficial species. Because while phytoplankton in general are very beneficial at the base of the food webs and support ecosystems, the harmful species can push out the beneficial species and disrupt these ecosystems. So it'll be interesting to see how the harmful species compare to the beneficial species and how this is changing with the climate and nutrient pollution. So I'd like to thank my mentors, Ellie and Carol, Dr. Joanne Burkholder, and the Center for Applied Ecology Lab for all the support. Thanks for listening. Do you have any questions?
I have an ignorant question about machine learning.
Do
you dictate that temperature is the first variable considered, or does Wika figure that out?
Yeah, so it's classified by season, since like phytoplankton varies seasonally, but no, it that was produced by Wika, so it identified temperature as the overarching factor in like that decision tree I showed.
I was curious if you have a sense of what impact a hurricane or tropical storm, can you account for that in the data, or can your machine learning algorithm?
Yeah, so It's in, so we like compiled a large amount of data, so obviously those impacts are gonna be in the data, but since it's over such a long period of time and we're controlling by like season and classifying by season, I think those are just like natural things that will show up in the data over time. But yeah, probably like if increased hurricane incidence is happening, that's probably also gonna show up because there's greater effects from that.
You include them?
Wind?
Yeah, when might we need to add because of that point?
Yeah, no, we haven't, I don't think so.
In other words, this is really step one.
Yeah.
There's a lot more you wanna
Yeah,
do.
there's a whole bunch more that we could do with the machine learning and the different factors we could add in, so.
Yeah, so some ignorant questions to be uninitiated. Could you briefly describe what a random forest
Yeah,
is? And could you also describe a little bit about what these algae are and what they do? And the one that was the GYN.
yeah, genotype.
Just tell us a little bit about what.
Yes, so the different algorithms I used were a combination of neural networks and decision trees. Let me see if I can't go back. But there.
Oh, there you
So yeah, random forest was the one that performed the best. And I believe it was a decision tree. So it operates in that, like how that graph I showed, which the machine learning basically branches off based on the different factors and then groups together based on how the variables are interacting. The neural networks try to like model how the brain works in some ways, like, you know, how like the neurons connect together. So the computer kind of mimics that. The different species, yeah, I don't know much about like each specific species. But yeah, do you guys want to say anything?
G1 mentioned Dave that Gymnodinium hetero capsa probably is there. I'm not sure about the T. And Procentrum minimum, those are all dinoflagellates, and hetero capsa triquetra can be harmful. Procentrum minimum can be toxic. Gymnodinioids are a group. They include some toxic, some non-toxic. They're all dinoflagellates. And dinoflagellates are very interesting. They can photosynthesize while they're acting like miniature fetus fly traps, and they're capturing prey. So they're not just photosynthetic. And then the oxyrus is a primitive pre-dinoflagellate. It's on the bottom left there. And oscillatoria is on the bottom right. That is a cyanobacterium, which can also be toxic or harmful. I'll leave you whatever.
Yeah, and we thought it was interesting that these cyanobacteria are negatively correlated with some of these nutrients. So the algorithms are pointing out these relationships as well that's important.
So while most of them are positive, there's a couple that are apparently being driven out by the guys who are stimulated. Because I say that because oscillatoria, the other one that looks like it, they're the second from the top of Vanazamenon, normally are considered nutrient loving, but the dinos are kind of, they kind of rule the estuaries for nutrient pollution, and cyanobacteria rule freshwaters. They're probably not doing as well as that. That's a mouthful.
But PowerPoint captioning picks you up a lot better than it picks me up.
Well, I don't know if it's a question or more just you talking about a lot of next steps. It'd be really interesting to take the relationship that you're kind of characterizing here. And if there were fish kill data to kind of test for correlations there, because then you could maybe ultimately predict when your risk level for a fish kill is very high, but I don't know how well those data are being collected.
Yeah, another interesting thing would be like to overlay it in like a GIS program, like overlay some of this data, and like if you had fish kill data to overlay that in a GIS program would also be pretty interesting.
A risk assessment tool.
Yeah. Yeah.
Fish kill data are no longer being collected by the state. They had a fish kill rapid response team and they axed it about 15 years ago. Give it citizens report, you might get, you might get, not get somebody going out, but there used to be a fish kill rapid response team and a fish kill day.
Sarah, thank you so
Yeah,
much.
thank you.
Hi, I'm Maggie, and for my project, I looked into the effects of shrimp on controlling macroinvertebrates under drought conditions. And so a little bit of a backstory here, the Caribbean is experiencing more low precipitation trends, and you can see by this graph. We're predicting from climate change predictions, they are estimating a 10% decrease in the amount of precipitation in that region. You can see here as well, the 10% decrease annually. So over time, more droughts will become frequent and stream ecosystems will have more periods of low flow, as well as there's unknown effects on the ecosystem. In the ecosystem, the one we're going to be focusing on today is freshwater mountain streams. And the dominant major consumer in the ecosystem is these freshwater shrimps you can see right up there. And they kind of eat basically everything in there. They eat leaves, algae, all the macroinvertebrates. So they play a big role in controlling that ecosystem. The study, that's right. Okay. So for the objectives for this were to take advantage of the ongoing flow reduction experiment through the lab to look at how the shrimps are impacting the macroinvertebrates under these low flow conditions. The study site we're focusing on is El Yunque National Forest in Puerto Rico, and the stream splits into two tributaries, Prieta A and Prieta B. These are first to second order, and you can see in this picture here, they're dominated by boulders and cobbles, and surrounding it is a dense riparian forest. The stream itself has variable discharge, so there's kind of no period of drought or flooding season. It's kind of unpredictable throughout the year. The ongoing flow reduction experiment, this little bit of backstory on how this works. The red line you see there is following this pipe that redirects the flow of the water around the stream 130 meters down. The water enters through that box up top and it follows down through the pipe. This net here catches any leaf litter so it doesn't block up the pipe. And this barrier you can kind of see here, the wood, anything that goes past it is what flows down the stream into the channel. So the pipe will just redirect some of that. The one thing to mention that's really important though is that this barrier cannot control for flooding. So any major flooding that occurs, it's just the flow reduction is not going to be effective. So this graph, we can see that this black line here, it represents like the water that's going down the channel of the stream. So anything that makes it over that barrier that we just showed. And this blue area is when the flow reduction is applied. The blue is combining what goes down the stream as well as what goes through the pipe. So kind of like what would naturally go down the stream if there was no flow reduction. And the black is with the flow reduction. So you can see that it is effective at decreasing the amount of water. In each stream, we selected 3 pools, and per pool there's 2 treatments. One excluded the shrimp using electricity, and one as a control with the shrimp present. And this was so we could see what effect the shrimps had. This experiment for the electrical exclusion was ran by the summer REU students, and their samples were collected, but no project was done with those samples, so that's what we're looking at now. So the way the electrical exclusion works is that fences produce pulses of electricity under these tiles that you can see. And this affects a small area of the stream to stop the shrimps from foraging in that area. But this does not impact the small invertebrates that we're looking at, like microinvertebrates. And this is just a closer look at what those tiles look like. So one will be electrical exclusion and one will be no electricity, just to control. The shrimp populations are also part of a ongoing assessment. They're baited into these traps that you can see here for long-term monitoring. So we can look at some of this to understand kind of how they're being impacted by the low precipitation. So the capture per unit effort is shown here in the two streams. The natural flow stream has more shrimp present than the low flow stream just because of the general makeup of the two streams is different. However, they remain consistent even when low flow is applied, which is the important part. So there's no real effect to the shrimp by the low flow. And then here we can see the effect on the macroinvertebrates. So up top here is the abundance of macroinvertebrates studied under natural flow control, natural flow exclusion, and then low flow control and exclusion. And the only significant difference is seen in the low flow. There's no significant difference in the natural flow. And same down here when we look at species richness. There's no significant difference in natural flow, only low flow. And we also looked at for two of the more dominant feeding groups, predators and collectors of the insects, and similar results were seen, that only the low flow saw a significant difference. But this could be due to the fact that the natural flow stream is much narrower, so when the flooding does occur, it's more intense, and that could be impacting this. Okay, bear with me here. This is a little complicated, but we'll get through it. This is an ordination analysis of those macroinvertebrate assemblages and what they're made of, their composition. So each point with the letter and number units represents a rep in our experiment. And the closer together those reps are, the more similar the composition of that assemblage is. So this first one here is the natural flow control. See how they group together, kind of similar. And then the second one is a grouping for the exclusion and the natural control. And you can see that they're pretty similar, they're pretty close together, other than this one outlier we have right over here. But for the most part, there's not much difference. And then when you compare the two controls between the low flow and the natural flow, they're further apart because, as we talked about previously, they have different composition in the two streams. And then we can look at the low flow control and the low flow exclusion, and they are more far apart, showing that their compositions have different makeups with the shrimp present or not. And so kind of the major finding we found is that under the low flow conditions only was the shrimp presence important. And this can be applied to understand the implications of how the climate change information we discussed at the beginning will be impacted in these ecosystems. And future research, I suggest, is looking down that food web and seeing how algae and leaf litter might be impacted by the shrimps under the low flow. And then I'd like to thank my mentor, Dr. Ramirez, and everyone in the lab, as well as the summer REU students who did the electrical exclusion. And any questions? Yes.
Shrimp have a higher impact on macroinvertebrates in the low flow, because there was like less availability for like what they would crave on to disperse and go to different areas.
Yeah, so that could be part of it. It also could be the fact that like with less flow, the pools are more like stable, like they have no current coming through them kind of. So that could allow the shrimps to like impact it better. They're not moving around as much with the flow. Yes.
You know why the electricity doesn't affect the smaller invertebrates?
That's a good question. I think it's just, and Dr. Ramirez can chime in if I'm wrong, but I think it's just the fact that the currents are so small. I'm not sure.
Yeah, most of the little ones are soft bodied, so they don't have a backbone or a camera or anything to conduct the electricity. So if you stick your hand, you feel the pulses. Fish will swim away, and shrimp also feel the pulses. But anything smaller than that, they don't. That's traffic and
All right, thank you so much.
Okay, I'm David. I did my research on macroinvertebrate education analysis. Just a quick look at my outline here. I'm going to talk about the triangle. It's a abstract concept that me and a couple of classmates came up with to kind of describe what it means to be an effective science communicator. I'll give you an example of what that looks like, and then I'll go into my research question, how I want to use this triangle that I'm talking about to address what I'm seeing as a pitfall with macro education. And of course, if I'm addressing a pitfall, I'm going to need to talk about how I addressed it and what my results were. And I'll kind of conclude with further recommendations or further research going forward. So the triangle. Here we can see it's an equilateral triangle. This is to suggest that each one of these parts is one, not more important than the other, and two, there's no specific order that it happens. All three of these things are present in an effective science communicator, and it's something that they interacted with at some point of their life. I'll give you an example of what this looks like. So in my example here, we have someone taking Clyde Sorensen's Insects and People class. This is a business major, and this is not even remotely adjacent to what their career aspirations are, but they find themselves sitting there and being captivated by Clyde and just the content of what macroinvertebrates are. After doing this, they start engaging in the media a little bit more. Now their TikTok feed, their YouTube, whatever it is, what they're doing in their personal time is starting to reflect this interest that they have, this growing interest. And from here, let's say they take it upon themselves. They go out and they want to test what they've learned. They've had this niche information given to them, and they go to their local park, catch some macroinvertebrates in a net, and see if they can identify them. You can see how any one of these three things could have been where it started or ended, but the point of it is that it's cyclical. The access to information that's interesting and engaging kind of encourages you to keep going and finding more of it and building upon it. So I said that there was a problem here. Where is that problem? Well, giving more context now to unique experience, education, and communication, I'm saying that both education and unique experience are lacking. For one, in the example that I gave in education, specialized macroinvertebrate education is generally walled behind secondary education. While it is a topic to learn about macroinvertebrates as bioindicators, you don't have the time to learn about a semester of some of the novel characteristics of specific taxa. Further, the example that I gave is not really accidental. You don't find yourself in a macroinvertebrate class as a business major unless you were trying to get like an easy A in Clyde's class. For the most part, people that are taking these classes are already at least adjacent to that interest in their career path. With unique experience, it's not always feasible in primary education. As I said, it is a topic that's covered in primary education, but if it's only one day, making a full field trip out of it is not time efficient or financially efficient. But going forward from that, when we have this perfect world where you are this business major and you do find things interesting, you're engaging in media and you're developing this interest, you still have to A, have a place to go to have this unique experience, and B, know that it's there. So going forward with these things, I have my research question and how I investigated developing this research question. How can non-college macroinvertebrate education be tailored to make more science communicators? Well, my targeted group was going to be early high school students with little to no access to unique experience. These are people that are learning about it in 8th to 9th grade, as it is an education standard, but also don't have this access that they're just going to walk into a park and then find themselves there. Most of these kids are going to be playing sports, having a local gathering place, or just maybe getting online and playing games at home. For preparing, I decided to assess where I work at Walnut Creek Wetland Center. We have current macroinvertebrate education, and I want to see what they were doing well and what they were not doing well. After doing that, I put together a class going off of these things with a classroom session to show fundamentals in an engaging way, kind of giving credence to what makes macros cool and also just the word itself. And after that, I went with an outdoor session to give context to those fundamentals and give a unique experience to crystallize this information and give context to what we do with it. So for my methods here, how did I change the lesson? I talked about how I looked at it. Well, to start, Walnut Creek Wetland Park's hand-on activity is a mock macroinvertebrate survey. Essentially, students will have a bin in front of them and they'll have colored beads. They'll sample from that bin, and each bead color is associated with a different macroinvertebrate. They'll look at a sheet and they'll be able to say, okay, I got X amount of dragonflies, X amount of caddisflies. Why don't I calculate my tolerance values and have a index to say what my stream quality is? Well, sitting down in the first time, it was very engaging. Students really enjoyed this. But I found that two things were occurring. First, the bead activity ultimately became a race every time. It wasn't about, wow, I love macros. It was, how fast can I get my beads before my friend? So I was immediately just seeing this divide between engagement and subject matter. They were interested, but it wasn't about the topic. And then going forward from that, after they did that, there was a very big dip in engagement because that got into the arithmetic side of things. They now had to calculate this biotic index with a tolerance value per order, how many of that order they found, and dividing it by the total number. It took them a couple minutes. So for my group, I wanted to get rid of the arithmetic part of it to kind of further separate it from this traditional classroom education. And then on top of that, I added methods of collecting macros and the respective habitats that they could be collected from, because I wanted to give them this opportunity to engage in this unique experience after the class, while also giving them the knowledge they need to do it effectively during the class. So how did I measure the effectiveness? I say that I'm changing it. I tell you how I'm changing it. I used visual observation of engagement at different times of the session, and I also gauged retention between the two by having qualifying questions. This visual observation is just, I'm at this portion of the section, I'm seeing that you are paying attention, I'm seeing that you asked me questions, we're engaging in discussion, and I just leave a log of that. As far as the qualifying questions, I'll get to that later. But for my first lesson, we're in the classroom. I ended up working with Peach Road Cultural Center. They had an after-school program where students between 9th and 11th grade would show up after school and they'd meet there and congregate and have activities. I had access to that because it's more or less a sister group to where I work at Walnut Creek. In my first group, I had 7 9th grade students. So right away, the location is great. They don't have that unique experience as readily available to them because it's kind of well nested within the city. And then for the age group, it's also good because it's an education standard at 8th and 9th grade to be learning about macroinvertebrates as indicators. For my lesson plan, my intro started with, again, giving this backbone of these creatures without backbones, of what macroinvertebrates are, why are they important, what is a tolerance or intolerance to pollution, and all of that was to give a backbone to the topics to follow suit, which was the use of ArcGIS. What do people do with this information? And why are these scores low around here locally, but not in the mountains? Giving them ideas to think about. And we closed off with how to catch macros, where we find them, and what streams they proliferate in. For my second lesson, we took the kids out to Walnut Creek. And interestingly enough, it's only 2 miles away from Peach Road, but that is a substantial distance when you think about it. If you don't have the ability to drive, you're not just going to randomly find yourself there. So you do have to know that it exists and then ask for permission for your parents. Point is, this is a unique experience. This is close enough to be available to them, but not close enough for them to really stumble upon it. For my group, I had 12 9th graders, so almost double the first time. This is probably because it was a field trip, but it was still great. As you can see, everyone's in ponchos. It was a downpour, and we did have to walk across the bridge of, are we actually going to be able to collect today? And everybody wanted to. For our objectives, I wanted to challenge the students to find macros using the methods that we talked about in class. We use DNets primarily, and we put them into bins to sift through and ordered them by taxa within an ice cube tray. Also kind of helps so that they wouldn't fight each other because a lot of the caddisflies do that. We asked students to identify finding their finds with an identification key. This is also kind of going back to this qualifying, did you take in the information that we taught you? And then recalling that ArcGIS, okay? We see that there's a lot of red around Raleigh. This is the number that we would give for red. What do you think this particular stream was? So for my findings here, this is just a graph going over start of my presentation to end. And also this is the order of things occurring. And on the left side, you can see the students' engagement. Right off the bat, giving them these definitions and all this jargon about what a macroinvertebrate is. This is a caddisfly, this is a dragonfly, what is the difference? This had the least amount of engagement. And I think with any science communicator, you run into this issue. The front loading of giving this information is oftentimes the first part where people start kind of getting dejected and like, I don't know that I can really have the mental strength for that. But then ArcGIS, wow, this part I wasn't actually interacting with them too much. I was kind of just demonstrating and giving the idea in their head that like we have a way to use this information and develop some sort of inference from macros in a stream. After that, we had the interactive portion. In this portion, I'm saying, okay, like you, what is an example of a place you've been to? Yes, let's look that up. Or do you think this would be worse or better than this one? Well, let's go see. Make your bets now. All of this was just getting them to critically think about, again, what we learned in the classroom and how it affects real world context. At the end, I did where and how to catch, and this was a similar vein. It was interactive as well. It didn't hinge on it as much, but the gist of it is, all right, this is a picture of a stream. What do you think we would find here? And we would go forward from there. So for the lesson 2 objectives, these were the qualifying questions and things I was looking for to see how effective it was. What would you rate our stream? I had 7 out of the 12 kids answer, and 3 of which landed between 5 or 7, which is like 50% of the people that answered, and that is about where I wanted them to answer. You know, the day that we had about 5 representative taxa between the crayfish, damselfly and dragonflies, true flies, worms, and a mollusk. We found a fingernail clam. That was cool. But kids actually able to identify macros using the key, 6 out of 12. They didn't all have a key. Some of them didn't want to use the key, but the ones that wanted to, they did it. 6 out of 12 were able to identify to order. And 11 out of the 12 actually caught a macroinvertebrate. It was a prolific day for dragonflies. Basically every kid, but 1 caught a dragonfly. They're everywhere. So for closing, my recommendations, just kind of taking all this information and what I think should go forward with macroinvertebrate education. So pairing education with unique experience. I had a lot of success with the interactive portion, but far and away, the most success that I had was actually getting them out there and getting them to collect macros. I had kids that didn't know what a dragonfly was a week ago, or at least the difference between that and a crayfish, up in arms about finding one and not telling their friends that they were the ones to identify it. Like, don't tell them. And they're like, okay, I'm the smartest person ever. Finding ways to incorporate this interaction. Like even with something with ArcGIS, I was able to get kids to talk about something they didn't know about 10 minutes before and make it exciting because there was this interaction. And then providing real world context. Again, this is hinging on ArcGIS. I was very pleasantly surprised to see how effective it got kids to be thinking about this stuff. And the reality of it was that I just showed them what you're doing with this abstract information. It has a tolerance value of 6. Okay, what does that mean? What does that look like? And then tailoring the method of delivery when applicable. This is really important. It's just a common thing to know your audience and what you need to be saying, how you need to be saying it. And it's very important with science because things get jargony. With this particular group of kids, I had my expectations managed severely by their activity coordinator, saying that they weren't going to be able to engage very well. They normally get distracted. They normally come to the center to play on the PlayStation, etc. I scrapped a lot of work to get to what I did with ArcGIS. Going forward with further research and improvement. Small scale, the obvious one is a survey before and after lesson to gauge affect and retention. That wasn't something I was able to do because there's training and permits involved with doing that. And you can see an obvious success with that. Me saying you're engaged is a lot more subjective than them actually saying I was engaged. Going forward with that, providing a third class with pristine conditions to contrast the first outdoor lesson. The first one, since it was Walnut Creek Wetland Center, it was actually a miracle that we found as much as we did, but there would be a great strength to having one like maybe the Eno River to also have to compare, because what they learned in class is not fully reflected by my first lesson or outside. For large scale, a longitudinal study following students exposed to science-rich primary education. This is more to strengthen the hypothesis of the triangle, but it still works for my research question regarding how to make the education more effective if the way that we're gauging that effective education is how many people join that field. But by looking at people over time, we can see, okay, well, they actually were X amount more likely to take part in this because of this effective science communication that they were exposed to early on. But again, I didn't have 15 years. And with that, I close off. This is a group, the group here, and they were very excited for the prospect of having a picture. And that was my caddisfly that I made on Canva for free. that took like 2 hours. But yeah, thanks. Questions? Yes.
Were you able to identify any specific factor that drove their interest and motivation to go out? I mean, I think it's so impressive that, especially if they're documented, like the students' interest is typically to engage with video games, that on a rainy day, they're all gung ho to go.
Yeah, the thing that I kind of identified was just actually being able to talk with the kids and have a fun time. This player here is a wrestler in high school, and I did that in high school, and they actually helped me so much with getting everybody else involved. The first day it was just them. The second day there was 8. The third day there were 12. I think really just engaging with them, like before, during, and after the lesson, making me look like someone they want to talk to and experience this with, was a big factor. Because I didn't really have as much trouble as they told me that I would have. We actually all had a fun time.
I have more questions, but
I have a question. Can you tell us a little bit more about how you measured engagement? Because I'm guessing that those different phases, engagement looks different.
Yeah. So again, I say visual. It definitely depends on person. This is one of the shortcomings of not being able to do a survey. But for example, with the ArcGIS interactive portion, I'm able to ask everybody in the room to add something on it. Not saying anything doesn't mean that you're not interacting or that you're not paying attention, but I would say that you're not necessarily engaging with the content up front. So I would say with something with interactive, it's a bit easier to make that like differentiation. But in the parts where I'm just reading off a screen and telling you this is a caddisfly, this is a dragonfly, interaction at that point or engagement would be just eyes on the board or raising your hand, which is a step above that. But again, front loading information, it's kind of hard to get that question in. So I would say it was kind of just off personal judgment, which is why I think a survey would be really good, because you could actually get them to say that, oh yeah, I didn't like that, or I did like that.
Lots of interesting ways to think about that from the science ed world, about like interactions between students with each other and with the instructor.
Absolutely.
And there's this interesting instrument, the teacher belief interview, that tries to conceptualize what you just explained, like how teachers try to use visual cues in the classroom. But I thought that was really interesting how you were trying to track that engagement over a number of different things. Thank you.
Yes.
I guess based on that engagement, you noted that it was kind of low at the beginning. Would you recommend, or are you interested in like changing the order or the type of activities that you would present to try and like get everybody really, from the first?
I mean, obviously this is something that's not unique in any way, but a lot of people and a lot of educators like starting with icebreakers, having something that's just like, name your dog and then 2 truths and a lie, something like that, just something like that, but more geared towards education. Even within the facility, we have educators that do things like this, where it's a stream activity and you're going to be a dragonfly and you're going to be a caddisfly. We're going to do X thing, something like that. Just some sort of interaction. In my notes that aren't really covered in here, what I wanted to do if I were to do it all over again would be trying to find some sort of way to interact through all of it, or starting kind of flipping the order of it. The reason I ordered it the way that I did was because being able to know what a tolerance value means, first, you need to know what the things are. So there's kind of like a constraint on the order of the content, but the way that you frame it would be how I would change it. I would say the content, if it were more interactive, there could be more engagement. Yes.
And her school has like a STEM chair position. And I think that the teacher was like more ecology based background. She was able to take like some of the older students to a stream, local stream, and they sampled for microinvertebrates there. So
That's awesome.
I think that's very good.
Yeah, absolutely. And again, like that triangle, that unique experience that they have, then they're going to have that edge when they get into the classroom at 8th or 9th grade and they're talking about macros. So like, I remember that, and maybe that's that spark that gets them to look back into it in their YouTube feed, and they get to the end of high school and they're like, man, I really like entomology. And then they do get to secondary education, or they don't, but either way, those things build upon each other.
Okay, one more thing, since you said survey was really hard, and I totally get that. I wonder if you could have like a tag for social media. And that could be a way of gauging communication. You know, if they tag the Walnut Creek Wetland Center and have hashtag macros or something. That could be a way that you could even informally, less formally gauge like how many tags are posted. I'm botching Heather's language now, but you know, a way to gauge engagement beyond your activity and communication, like with their perceived peers.
Yeah, I think I've indirectly done something like that, since the place where I work at Walnut Creek is pretty close knit in terms of like, oh, hey, we saw this person today. I've been able to actually hear about a lot of these kids coming back and engaging in it more from some of my coworkers. But that is a good point. That's like a self-report sort of thing that isn't like particularly taxing on the individual. I think the only thing I might run into there is like a non-response, where when I say, here's a survey, I'm probably more likely, but then again, because I'm saying here's a survey, there's more things involved. So yeah, I think that is a good like route around that issue. Yes.
Stuck out to me as you were explaining your project is how important the rapport is with the students, and then two, how pessimistic the instructor was.
Yeah.
So I think if you were to continue this, you should like continue to involve what you do with the students, but you should create something to give to the instructors beforehand to get them on board, because clearly they also need some science education.
Yeah, it was actually perfect timing that since it's a cultural center and not a community center like at Walnut Creek Wetland Park, their priority of activities are like, some is like culinary, some is like stitching, some of it is history, but most of it hasn't actually been science. And so when I was reaching out to kind of find somewhere that I could do this through Raleigh Parks, they were looking for science education. Literally, their organizer was a month or two into the process of trying to develop one. So we just like met paths at this time where I wanted to do something and they needed something, and we did it. And now we're working with that park actually as a sister group, even more so than I said before, to get a recurring group coming back, which was really awesome because I was not expecting it to be that effective. But yeah, I think just meeting the kids beforehand and like talking to them about it got them the most excited. But yeah, I did get my expectations managed a little.
You just see the instructor like skewing your data because, or like changing the experiment because if they're making comments in the van on the drive over, you know what I mean? They're
Yeah.
setting you up for failure. So
Yeah, like, all right, guys, you need to pay attention. It was like, I was gonna pay attention. Like, maybe I'll kick rocks. I don't know. Yeah.
Thank you so much. Thank you.
All right, hi everybody. I'm Chloe with the Hopkins Lab, the Sustainable Health and Ecology Lab. Today I'm going to be talking about snake parasitism occurrence in intermediate hosts. So all vertebrate species have at least one parasitic worm species that's been studied, but only 50% of snake species in the US and Canada have one recorded species. So our lab researches parasites in snakes to better understand the patterns of parasite diversity. For example, in the snake species that we've surveyed, we've observed that some snake species have high parasite diversity and most individuals are affected, where some species may not have a ton of individuals that are affected and they don't have a lot of diversity. Oh, it appears my speaker notes are off. The speaker notes are off.
This is a comment from.
This should be on the previous one.
I don't know. Sorry.
Okay.
I downloaded it from the
Yeah.
folder.
Okay, you okay? Yeah, I may just have to do a little bit more back and forth. But as in this one, wait, oh. No, okay, it's right. I haven't used PowerPoint that much. There are many different types of worms in our snakes, which may be present in adult or larval stages. The trematodes are flatworms. Those are the ones in the top left, which are the ones that I'm going to be talking the most about. Then the nematodes below that are roundworms, which are also super common. Acanthocephalans are thorny-headed worms, tapeworms, and then pentostomes. Those ones are not as common. Those are found in the lungs, but just focus on trematodes and nematodes for this one. Most of the parasitic worms that we find have complex life cycles where they infect different host species as adults and in larval stages. Snakes usually become infected through trophic transmission. So they consume something that already has a parasite and then become infected themselves. For example, this one starts out, it's a trematode life cycle, the flatworms. It starts in the larval stage in the water, will go to aquatic snails and then can go to the fish as well. And then it reaches its final life stage inside the snake, which is the final host. Oh, and the ones that we see in the juvenile stage that are found in like the fish are the metacercaria. So it's like a trematode, but in a little cyst. It seems like snakes with different diets have different parasite communities, and that's something that our lab is working to document. In order to do so, we need to understand what is in the specific prey that each specific snake species eats. The overall goal for my project was to start documenting the parasite communities of the hosts. I looked inside roadkill, frogs, salamanders, lizards, toads, basically whatever we could get our hands on, and primarily fish that were found dead near streams or ponds. We only dissect what we have available and things that have already passed. So this graph shows what we've found so far. What you can see is that bluegill and largemouth bass, both fish, are the highest amount of hosts. That is mostly because that is the most samples that we've had, but we do have a pretty decent diversity of other things, frogs, salamanders, and lizards, but fish are the most common ones. Most of our specimens come from 2 areas within North Carolina where we have people out looking for dead animals to collect, DOR, dead on the road. And each star on this graph that I made shows a location where a specimen was collected. So we have a lot right up in Wake County and Durham County, but then a lot near Bladen and Sampson County. I spent most of my time dissecting juvenile bluegill and largemouth bass from a large flood event in a Wake County pond after a storm. Today, I'm going to be sharing what I learned by comparing the parasite communities of these 2 common fish species, which are great snacks for snakes. After thawing out the tiny frozen fish, we dissected out their organs and then squashed the organs and the rest of the body between 2 large glass slides. I'd then go under a microscope and push it down so that way I could see through everything and look for parasites. I would circle and then count any parasites I found. Sometimes they're super duper tiny, given that most of our fish were like 2 grams and could have up to like 80 parasites in them. So we would use a compound microscope. On the left, that's about the size of the average fish that I was looking at, so they're very small. Like I said, we did get some other samples, so that second picture is me dissecting a species of catfish that we were able to find. So that one was much bigger. These are tray about this big. These are the catfish gills. Usually they're much, much smaller and very hard to see. It's also hard to see here, but there is a little bit of Sharpie. So I would go and circle what I found, count it up, and then our PhD candidate, Alex, or my PI would go through and double check. And then these are some of the parasites we found. This one, there's actually 2 nematodes in there. This is that trematode metacercaria, the flatworm that I mentioned earlier. This is another nematode, but it's in a juvenile life stage, so it's in a cyst. And then this one is one of those thorny-headed worms. We found several species of parasites in each fish species. We haven't been able to identify them all exactly to species yet, but Alex is working on using genetic sequencing for identification. Most of the parasites were larval stages. These are all the ones that say something besides adult in parentheses. This means that they can go to infect predators like snakes, but interestingly, we didn't find acanthocephalans, the thorny-headed worms, in the largemouth bass, and we didn't find monogeneans, which are a gill parasite in the bluegill. The most abundant parasites were those trematode metacercaria that we had mentioned earlier, especially in the bluegill, and then adult nematodes in both the fish species. The other parasites were relatively rare, and we do see the differences in the species that I had mentioned with the acanthocephalans, and then the J3 nematodes, which were the ones in the cysts, and then the cestodes or tapeworms were only found in the bass. This graph shows the overall number of fish of both species that either had, or fish parasites of both species that either had no parasites or had one of the parasite taxa. Again, you can see that nematodes and the trematodes specifically were the most common parasite. In these two fish species, we found several parasite species, some of which were shared between the two host species, shown in the both, but then a couple of them were just in the bluegill or the largemouth, like shown in that pie chart up there. Of course, this study did have some limitations. It was a sample from just one pond at one time point from one large rain event, and we only sampled juvenile hosts, given that they were like 2 grams. Because that's what we had found. We did sample around 30 individuals of both species, which is a pretty good sample size. But it would probably take a much larger sample size, including different age ranges and locations, to observe every parasite species that infects the host specifically. All of this illustrates how hard it is to understand parasite diversity. It's a lot of work to thoroughly sample hosts. This is an ongoing effort for our lab. We plan to continue increasing our sample sizes, including taxa that we haven't surveyed yet, and new locations. But even when we find parasites, they can be quite difficult to identify. The larval worm on the bottom right had a lot of identifying features. So these are both nematodes, but one of them, it's got a thorny head, it's got more structures. This one's just clear, and it's much harder to be able to identify what it is. So it does require genetic sequencing. We are trying to identify more tools to quickly and easily identify parasites in our local wildlife so that we can understand how prey and predators are connected through parasites. So this is the Sustainable Health and Ecology Lab acknowledgements. My PI is Dr. Skylar Hopkins, Alex Nelson, and then my host project partner who's going to continue this research with more of the frogs, lizards, and more samples is Jake, and then we get specimen sourcing from these folks here. Thank you guys so much. Any questions? Yes.
So a lot of the fish types like really small, like babies. I was wondering if you think that the community of parasites in the fish might change just the fish get for.
Yeah, definitely. So they get the parasites either just from being in the water or from what they eat, and their diet is going to change as they get bigger and can fit more things in their mouths. But yeah, I feel like the stuff that I found was mostly like, we can apply it and it's great, but we can only apply it to this very small age group and specific species. But it's a step in the right direction. Yes.
As a piggyback, does that mean that those juvenile fish, because they're smaller, are they a potential food source and vector to different types of snakes than the larger adult fish would be?
Yeah, for sure. Most of the time we focus on Nerodia, which are just overall water snakes. But there are different, there'll be different prey as they're at different life stages. No other questions? Yes.
How big an impact do you think these parasites have on the snakes?
It kind of depends. Yeah, it's really interesting because a lot of other people in the lab focus just on snakes. So some of the snakes that aren't water snakes are the ones that will find maybe a parasite here and there, not in every snake. And when we do find them, we may not find too many, but water snakes are known just for having a ton in there. For a bigger snake that only has a couple, then they're probably not going to be as affected as much. They're there, but it's not going to make too much of a difference. On a smaller snake that has 200 parasites, it probably wasn't doing so hot.
Water snakes?
Yeah.
So they have a lot of parasites.
Yes, we don't get to sample them too much because we don't
They're not as common as just the normal like northern banded and brown water snakes. But yeah. Yes.
You mentioned that the sample is kind of divided into two little sections.
Yeah.
So I'm here. Were you able to look at if there was differences in the parasite communities between those two regions and what that might apply for where else you may want to look?
Yeah.
State, find, and samples.
Yeah, there's actually a specific species of parasite. I can't remember, but it's one that Alex really likes. That the Strigia ones, yes. Those ones are just in the northern, like closer to here part. Yeah, those ones are really interesting. We also look at snails in the lab since they are one of those intermediate hosts. We don't find those in the ones in Bladen and Sampson County. I would say that's maybe the most popular one that we're looking at right now, but we again just don't have that many samples to get a truly statistically significant. We can kind of more say correlation versus causation at this point. Like we haven't found this here, but that doesn't mean it's not there. It just might be more rare there.
Thanks, Chloe.
Thank you.
Okay, hi, my name is Catherine Laufenberg. I'm a member of Dr. Sheth's lab, and today I'll be talking about how I used climatic niches to investigate changing alpine plant communities in the White Mountains of California. So this photo was taken by my mentor, Caleb Goff, in the White Mountains, and I chose this to showcase some of the habitat conditions that alpine plants experience. So the alpine is defined as the region on a mountain above the tree line. And this is a region with high UV exposure, cold temperatures, limited nutrients, and short growing seasons. So this plant here is Erigeron pygmaeus. It's a pretty low growing hardy species. And this species and other alpine species need to be able to withstand unique abiotic conditions as well as competition with other nearby plants. So for this reason, sorry.
Right here.
Oh, thank you. For this reason, alpine plant communities may be particularly vulnerable to climate change and thermophilization. Oh, yeah. Thermophilization is the process of cold-adapted species becoming less abundant and warm-adapted species becoming more abundant in communities over time. And research can tell us how these communities are changing over time and how we can protect them and other similar systems. So for my project, I asked the question, are alpine plant communities in the White Mountains of California changing over time and across elevation in response to climate change? I hypothesized that lower elevations contain more warm, dry climatic niches. That's represented by these red flowers and the warmer colored flowers. And higher elevations contain more cool, wet climatic niches. And over time, alpine plant communities are losing cold adapted species and gaining warm adapted species. That was my second hypothesis. And that might indicate that thermophilization is occurring in these communities. To test these hypotheses, I had to create community climatic niche means, or CCNMs. These are weighted averages of the climatic niche affinities of each species within a community. So each species is found within a certain set of environmental conditions, and we can average those environmental conditions and weight them by abundance to describe the entire community. So thankfully, we have that abundance data due to Dr. Sheth's participation in this project called Gloria Great Basin. It's part of an international collaboration to study the effects of climate change on alpine plants. And my mentor, Caleb, made this figure to show how the surveys are conducted through Gloria Great Basin. The White Mountain Range is made up of 5 peaks, each surveyed every 5 years since 2007. And within each peak, there are 8 to 14 100-meter long transects, starting from the top of the mountain until the tree line every 25 meters. These transects are broken up into 10 segments, and each segment contains a bunch of different plant species. And the way that abundance is recorded is however many segments a plant is present in within a transect, that's its abundance value. And for the purposes of my project, one community is equal to one of these transects on one peak from one survey year. So since we have all of this information about what species are present in the White Mountains, I was able to query the Global Biodiversity Information Facility, or GBIF, for 146 of these alpine species in the White Mountains. I collected 200,779 occurrences. Those are all of the occurrences. Each black circle represents one, and each occurrence is an herbarium record that contains information about the specimen, including where it was collected, so the coordinates. And that's helpful because I can take each coordinate, each latitude and longitude, and overlay it on top of climate data, and then I can assign climate values to each occurrence. So the map that you're looking at right now has a background of Bio 1, mean annual temperature in degrees Celsius. And I also downloaded 3 other bioclimatic variables from Chelsea Climate. So I also downloaded Bio 5, maximum temperature of warmest month, Bio 6, minimum temperature of coldest month, and Bio 12, annual precipitation in millimeters. So now I'm going to take you through how kind of like a dummy community niche mean would be calculated. So in a real calculation, there are a lot more species, but we'll just take 2 for now. So this is Erigeron pygmaeus. You might remember it from the first slide. This species has a pretty narrow geographic distribution. It's confined to the Great Basin, and each of those white circles represents one of its occurrences. So since it's overlaid on top of Bio 1 again, mean annual temperature, I can average each of the climate values that those coordinates are found within and get a species average of 1.1 degrees Celsius. And I can also do the same thing for annual precipitation. Erigeron pygmaeus has an average annual precipitation of 785.28 millimeters. And then this is Calaria macrantha. It's a grass species that has a much broader distribution. And I can do the same thing and create a species average for mean annual temperature. That's 8.03 in this case. And for annual precipitation, 577.34 millimeters. So next, now that we have the species averages, I can weight those by abundance. So here's our Gloria Great Basin transect again. Calaria macrantha is present in 7 out of the 10 segments, so its abundance is 7, and Erigeron pygmaeus is present in 3 out of the 10, so its abundance is 3. So here's the calculation that I would do to describe the niche affinity of this community. We have our 2 species values for mean annual temperature, and then abundance. Here's our equation. And so the community niche mean for mean annual temperature for this community is 5.951 degrees Celsius. And here's the same calculation, but for precipitation. You can see we have the same abundance values for this community, but different species means. And you get 639.722 millimeters. So I did this for a lot more communities with a lot more species in them. And I had to figure out how to analyze my results to answer this question. So I ran 2 linear regressions. Here's the R code for anybody who's interested in that. But each part of these equations corresponds to the question. So to figure out how alpine plant communities are changing, my CCNMs are the response variables, and the explanatory variables were time in years and elevation in meters. This is a fixed model that I ran for each peak. So I also looked at the interaction between year and elevation. And then this is a mixed effects model to look at all of the data, but with peak as a random effect because the data are not independent within peaks. So here are my results. Starting with elevation as a predictor, I'll take you through this figure. Each point represents one community, so one transect from one peak from one survey year. Each of these colored lines represents one peak, and that comes from the fixed model that I ran. And then this black line here shows the broader trend across the entire mountain range. It did not come from any of my linear regressions, but it's there for illustrative purposes. And as you can see, as elevation increases, the community niche for precipitation became wetter. So communities at higher elevations tended to have niches with higher or with wetter precipitation values. And it's interesting that these slopes vary within peaks, but it's kind of cool that there's kind of a general trend across the broader region. And elevation was actually the only significant predictor for niche means. You can see that same kind of situation here with temperature. So here's mean annual temperature, maximum temperature of the warmest month, and minimum temperature of the coldest month. We have the same setup, so each point represents one transect or one community. And again, the peaks have different slopes, but there's this overall trend of niche means for precipitation decreasing, or sorry, for temperature decreasing as elevation increases. So the second part was time. Time actually was not a significant predictor for these niche means. As you can see, each point represents a community again. The slopes don't really have a significant positive or negative trend within peaks. And as time increases, community niche affinities for precipitation stayed relatively consistent on each peak. And this was the same with temperature. Once again, I have mean, maximum, and minimum. You can see that there's no real positive or negative slope on any of these, or no significant positive or negative slope within any of these peaks. As time increased, community niche affinities for temperature stayed relatively consistent. So to go back to my question, are alpine plant communities in the White Mountains of California changing over time and across elevation in response to climate change? My first hypothesis was that lower elevations contain more warm, dry climatic niches, and higher elevations contain more cool, wet climatic niches. This was supported by my research, so elevation was a predictor for niche means, so that was an interesting finding. But maybe even more interesting, my second hypothesis, which was over time, alpine plant communities are losing cold adapted species and gaining warm adapted species. This was not supported. So that does not suggest that thermophilization is occurring in these alpine plant communities, but that could be for a few reasons. Maybe the communities are resilient to climate change, but I think more likely we don't have enough of a time frame to see the change because these are long-lived species on top of these mountains, and there could be adaptational lags. But overall, the findings are really cool, and understanding how niche affinities change across elevation would be super useful for future studies and conservation efforts. I think to see an effective time, it would be interesting to have more surveys so that we can have stronger findings. And it would also be interesting to ask the same question over a broader geographic range, like maybe multiple mountain ranges. I want to thank Dr. Sheth, Caleb Goff, Sophie Mang, the Sheth Lab team, and all past and present Gloria Great Basin contributors for their support. And here are my references.
You had said your second hypothesis wasn't supported. Was that because there was like, it's not that there was a transition from species, was it that the same species just kind of stayed?
Yeah, let me go back to that slide. Yeah, so that's true. The presence didn't really change much over time. And that could just be because they're super long-lived species and we're not seeing it because they're not dying yet or moving yet or dispersing their seeds? Yeah, good question.
So was it like you were seeing warmer species move in there too, but the same species that were there stayed, or there were no changes in the plant occurrences?
Yeah, we didn't see warmer species moving in because like that would indicate thermophilization was occurring and that wasn't happening.
Do you think that I'm really struck by like the high UV exposure. Do you think that that is going to be a significant enough selective pressure that it could like hold off invasion even if temperatures change?
That's an interesting idea. Yeah, I don't know. Maybe so. I mean, they're adapted for that specific environment, but then again, maybe warming temperatures would encourage more like lower elevation species to come up anyway. That's interesting. Yeah, there were certainly other factors that would be interesting to look into besides climate, like other biotic factors,
So you said that a lot of these plants are long-lived, given about how they live. How many years do you think you have to observe this be able to see change?
I'm not sure. Maybe, I mean, so we have about 18 years of data. So maybe double that and another 20 years we would, I'm not sure, see some change.
Thank you, Kathy.
Thanks.
Okay, hi everyone, my name is Andy. My presentation is called Dead and Alive, and I'll be talking about my experience and research with the mammalogy unit at the North Carolina Museum of Natural Sciences. Yeah, I've been working with them for about 5 semesters out of the 10 semesters I've been an undergrad here at NC State. So it's been a long time. I met Dr. Michael Cove in 2020 virtually when I was doing like COVID semester. And yeah, I've been with them ever since. So this is the collection. Took this picture because it kind of reminded me of like Night at the Museum. Like every time I walk by one of them, I'm like, oh, I'm like scared they're gonna come out and bite me. But yeah, I'm gonna talk about collections and why they matter. So collections hold a lot of historical data, especially like natural history data, which is really important. I mean, like even in the last presentation, like you used a lot of like stuff from herbariums. So this is like stuff that's definitely used to this day, like researchers that have come into the collection to physically look at these specimens and take measurements. But yeah, so the mammalogy unit has over 20,000 mammal specimens in its catalog. And that's just like the catalog. That's like not even like stuff that we just kind of have sitting like waiting to be cataloged. And a lot, the information for each specimen, they each have a unique catalog number, and they have data associated with it of the preparation number if we prepped it in the lab, or its original number from wherever it was like acquired from, maybe like a previous collection. And then locality information, that is a date, address, coordinates, or like from where it was collected and when, who collected it, who prepared it, who cataloged it, the species. And then if it's in the lab, well, yeah, you can get sex, age, reproductive status. And if it's in the lab, you can get measurements, weight, and tissue samples of the heart, liver, kidneys, and muscle. So this is prep work. I've been a part of most of the process, like the intake process for a specimen. So I'm gonna basically summarize that. So first you start with like the dead specimens. We have like some rats and mice over there. And then that's a picture of me skinning that, one of those mice. And that's like what one of the prep numbers looks like. That's from like a completely different specimen, but that's the skin that I did in that picture. And then one of those rats is that one right there. And then in the next picture, you'll see the body without the skin. Looks kind of freaky. But yeah, it was even worse with the rats. Like it was just like that, but huge. And this is, so after you skin them, the body, you take it to the beetles, and the beetles lay their larvae in the flesh, and then they help clean up the bones. And this is a seal that they've had in there like a while back. So for cataloging, this was like some of the first stuff I did when I got to the museum. There's this, they have like, I think they're on their 3rd edition of this like really giant, like these few giant books that they keep that have handwritten every single specimen in the collection. So I took that picture when I was doing some cataloging of, I think it might have been like, I think this picture is like one of the first few ones that I did. There were just kind of some random ones that needed to be put in. So yeah. And then, yeah, these pictures are like, show the bones of some of the really small stuff you can get. Like these vials right here, like can barely fit my pinky in them. So you can see like how small these skulls are. These are bat skulls. And then those are skeletons over there of some mice. And that is a Permiscus skull, I'm pretty sure. So you can see the teeth. The teeth are used for identification. So, you know, it's important to have that. And yeah, all those boxes I had to label, and I'm pretty sure those are all otter skulls. So you can get a sense of like, just a sheer amount of like stuff that there is. And yeah, this is like bigger stuff. These are some skins that I had pictures of. That is well, that's a fawn. And then that's a ocelot, and that's a red wolf, which I thought was cool. And I think those skulls up there are lions, and that's a beaver skull. So, oh, let's see. Is that gonna play? No. Okay. Well, it's a video of a bat flying. But so as part of my experience at the museum, I've had a few field experiences. My favorite was when we went out for one night and did some like, some biologists did like bat trapping of these big brown bats one night, and I helped with like data collection while they were taking measurements and all that and releasing them back. So that video showed like one of them being released, and you can see his wings flying in the air. It was cool. And then can't really see my face, but that's like a candid photo of a camera trap that I was working on. Okay, so for my research, I focused on the data the research takes place in Prairie Ridge Echo Station, which was once grassy fields, and then World War I training grounds, and then a prison farm, and then NC State University cow pasture, and is now a prairie and forest that's owned by the museum. And I feel like that bit of land has just like been through a ton, this little 45 acres. But it has all these features that make it like a hub for wildlife in this extremely developed area. And so the museum has been doing small mammal trapping out in Prairie Ridge since 2011. And a few years ago, they did like a preliminary study of the data where they found the density of hispid cotton rats using a spatially explicit capture-recapture model. And this is like a picture I took with my phone of a poster, so you can't really see the table well, but it's kind of like gives you a sense of what these were their results. And so in the tallgrass prairie of plot 3, there was the most captures, and they had the greatest estimates of about like 200 adults per hectare. And they peaked in the fall, and then they were lowest in the winter and spring. And my objectives, my goal is to like estimate the density of white-footed mice across the trapping sessions and compare their variation based on season. And I predict that the densities will be highest in the fall, similar to the cotton rats, and then lowest in the spring, and will have like a steady decrease over time, because that's what part of the reason why they really want to like analyze this data, because they've been noticing a lot less captures since they've started. So to do the small mammal trapping, they use Sherman live trapping and across 3 different plots with like different habitat types. They did it in a grid layout where there's about 50 traps per plot, so for 150 total, so it's a lot. And there's 1 session per season annually, like same month every year, same 4 months, and 4 trap nights for each session. And then the traps, cotton was placed in the traps and they were baited. They used to be baited with rolled oats and peanut butter, but like this picture I took, you can see that attracts a lot of fire ants, which is like awful for the any mouse that's gotten there. And so they've switched to corn and carrots because that still baits them and won't attract stuff. And this is what the plots look like at Prairie Ridge. The first one is a bottomland forest. Second one is fescue, which is like a cool season grass that's like green all year. And then the last one is plot 3, which is tall grass prairie, which is like prefers warm, so it's like dormant in the winter. These are some pictures I took of, these are white-footed mice from small mammal trapping, the times I got to volunteer. So for data collection, they get the number of the trap, the species, the tag number if they place one, or if it's a recapture, they'll collect that, the age, sex, reproductive status, weight, and then measurements. And they each get a permanent ear tag so that, like I said, like if you recapture them, you can collect that. And so I mentioned this before, the spatially explicit capture recapture. So this is like more preliminary data, like what I referenced previously, like what they did before. But what that is that it's a detection function that follows a half normal distribution, and it estimates density from 2 detection parameters. And so on this axis, this is an example from like a different type of model, but it's the same idea. The probability of capture is on this axis, and then the distance from the activity center, which is like this value on the curve, is like here. So it shows that your probability of detection is going to decrease in this pattern as the distance from its home range increases for the trap. And these are the results of what I did. So I kind of like put these dots here so you could like have an easier way of looking at what season is like each one. So it's like winter, spring, summer, and fall. So my prediction was like completely wrong. It actually was like the total opposite for the white-footed mice, which, I mean, I guess that makes sense. So kind of like in those first few, you can see that in the spring, there was a lot more captures, or a lot more higher density, and then it fell a lot more in the fall. And yeah, and I only have plot 1 and plot 2 up here because from that time span that I did this from like 2015 to 2019, only 1 Peromyscus was caught in plot 3. So like, obviously I couldn't do anything with that. Which was the opposite effect because the cotton rats were like most abundant, or most had the most density in plot 3. So yeah, I mean, population decrease of rodents will affect obviously other species. I think there's like 24 mammal species out in Prairie Ridge. So yeah, I mean, it'll have like effects across like trophic levels, and then like they're like system engineers, so it'll have impact on a lot of other things. For future research, there's some recent data. There was like a gap in the data from 2020 because of the pandemic. It was tough because, I mean, mice can get COVID. So there's been a lot more, I mean, there's like, the data from like 2023 and 2024 looks a bit different from the one from 2019. There's like more captures of the white-footed mice in plot 3 that I wanted to do, but didn't have time to do. And then I would like to do it as well for like the same model for this new time span of hispid cotton rats, which is the other species that they did before. So that would have taken a lot more time because, I mean, previously they had estimated like 200. So it was just a lot more captures, and I didn't have much time to do that. And then you can consider other variables in the model and create different groups. You could do it based on sex. And I could try like different models. I think the graphic that I had before with the graph, that was from like I think it was just a spatial one and not spatially explicit, which has like different parameters associated with it. So, or you could do like a robust model. Those sorts of things will give you different population parameters. Okay, my acknowledgements. Dr. Michael Cove is the research curator at the Museum for the Mammal Unit. Lisa Gatens, she's the collections manager. I've worked with her the most, and yeah, she's great. Shout out to her. And Dr. Macy Krishnamurthy. I've done a lot of stuff in the prep lab with her. She's taught me how to skin some stuff. Yeah, this picture is from like this is like a little news service. Yeah, this is me labeling a manatee skeleton that was like really big. That's Dr. Cove and that's Lisa. Are there any questions? Yes.
Sounds like you've had a lot of diverse experiences working in the mammal lab at the museum. I'm wondering if Dr. Cove has converted you to rodents, or if there's maybe another species that you'd be interested in researching.
I'm definitely converted to mammals. Like I major in engineering, but I want to study wildlife. And I've always kind of been unsure, but I mean, throughout undergrad, I've like more affinity for mammals now. But yeah, I mean, I've always really been interested in bats. So I would say like that's my favorite species. But I like working with mice. Are there any other questions? Yeah.
It said that you had found them like the complete opposite of what your hypothesis was. What were the reasons again that you thought that might have happened?
Just kind of generalizing it as like another rodent species. Like, I guess they could have like different behaviors around the traps, or just like they're probably not moving as much during different seasons. And so I assumed it was going to be the same for another rodent species, but it ended up being different. So yeah.
I'm just curious to know if like all of the collections data that y'all collect, is that part of like, does that contribute to a bigger like nationwide or like global database somewhere of like all museum collections? Or is it like, do y'all just have your data and like every museum has their own data separate?
I think they're all separate. Yeah, like they're all kept like in their own respective database. Like the Smithsonian has theirs, and then North Carolina has theirs. Yeah. Anything else? Okay.
Thank you so much, Andy.
Thanks. And thank you all again for coming. Really, really appreciate all of our students' hard work, all of our mentor support. Thank you. There you go. And thanks to Heather for this picture. So featuring wildlife here on campus along the Greenway. So if you are so inclined, a number of folks will be recongregating at Raleigh Brewing Company. You are welcome to join. If not, and regardless, happy end of semester. We have almost made it. And thank you all for your hard work. This has been tremendous. So thanks, y'all.